INU Smart Campus apps to check your attendance, and just enter 6932. Okay. So I will check it, the attendance right now. Okay. Lonika. So Lonika, so you had better open INU Smart Campus apps to check your attendance. So you had better enter 6932. Okay, Lonika. Okay.
Yes, Professor.
Okay. 신주환, 세무 회계학과. 지난번에 수업 안 들었는데 신주환 또 안 들었네. 열심히 한다고 해가지고 수업 들으라고 했는데 세무 회계학과인데. 오케이. Okay, so okay. Thibault. So I found that you didn't check the INU Smart Campus apps. So you had better open it and just enter 6932.
Yes, I'm doing it now.
Okay, it's good. Okay. Okay, now we will start our class. Okay, so last time I explained the the detailed information of the midterm exam. Okay. So do you have any other question in midterm exam? So now, do you have any other question in midterm exam? Okay. Okay. And okay, so once again, I will explain the procedure again. So the exam time is the October 22nd, Friday, next Friday, from 10:30. So you had better attend the Zoom around the 10:25 or 10:20. So I will check your status, whether your Zoom is okay or not. Okay. Okay. So the test time is unlimited. Okay. Take your time. So our test range is from chapter 1 to chapter 5. So today we will cover part of chapter 3 and chapter 4 and chapter 5. Okay. So important thing is you have to solve the test questions while showing yourself in real time through Zoom. Okay. And also, so after finish your midterm exam, you have to write and your name finished exam in the Zoom chatting window. So I will check. So after finished all of the exam, I will check this record, and also I will check this one. So you have to send it to my phone within 5 minutes. So I will check it, the time difference between the finished the exam in the Zoom chatting window, and also I will check the time you submitted to me. Finally, you have to make answer sheet as PDF version, and you can submit answer sheet through assignment in e-learning. Okay. Do you have any question in midterm exam? Okay. So because this is the midterm exam is taken by the Joon. So you have to follow this procedure, okay? So if you did not follow this procedure, I think that if you violated it, I think that you did cheating, okay? Okay, very important, okay? Okay. Okay. So now we will cover chapter 3 and chapter 4 and chapter 5. So do you have any other question in chapter 3 homework or chapter 3 the class material? Do you have any question? So okay, now we will cover chapter 3 again. Okay. So this is the second subject, heterochromatin in centromere. So in our homework, there was a question regarding the centromere structure. So centromere structure is composed of the central region and out region, okay? So actually the out region is the heterochromatin region, is composed of the OTRL and OTRR. It's like this, okay? And also we cover, I explained this experiment. So this is very important. So in midterm exam, this is very important. This is the experiment to reveal the function of the OTR sequence, especially the fragment DG. So based on this experiment, we can say that maybe we don't know, but some transcription factor maybe bind to the unique sequence in L5, maybe, and if that it will play an important role to form or heterochromatin. Okay, based on this experiment, we can say that. And also we cover some detailed information of the experiment to reveal that transcription in OTR region is important for heterochromatin formation. So we explain it. Okay, you will see that finally we find that so IBP2 mutant and IBP7 mutant cannot initiate the transcription in OTR region. So based on this observation, we can say that. So because this is the very important role in the RNA polymerase 2. So it means that the transcription in OTR region is important for heterochromatin region in the OTR region. Very complicated experiment, okay? Okay, but this is very important, okay? Okay, and also, okay. So this is from chapter 3, we covered RNAi dependent heterochromatin formation. Previously in chapter 2, we covered the birding yeast. So in birding yeast, there is no RNAi dependent heterochromatin formation. But from fission yeast and mammalian, and also all of the advanced organisms, they have the RNAi dependent heterochromatin formation. Okay. So this is the first procedure and second procedure and third procedure. And this is the final procedure is like this. Okay. So you don't have to memorize all steps. Okay. For example, in our homework, we mentioned the very important step in the RNAi dependent. Okay. Where is it? This is chapter 3, chapter 4. In chapter 3, we cover that. Okay, this one. So we cover that. So the following events are related to the model of early heterochromatin formation by RNAi and list the order of this event correctly. Okay, like this. Okay. So you don't have to memorize all of steps, but you have to know the procedure. Okay, so you have to know the important thing. Okay. Okay. The first step is that the function of exosome and the function of the treatment. Okay, so the bidirectional transcription happen in the OTR region. And also, especially the exon is remove most of the non-coding RNA. But so if the treatment is bind to the non-coding RNA, in this case, exon cannot remove that non-coding RNAs. And so finally, the treatment bind to the remained non-coding RNA. After they will make the generation of pre-RNA. They will make the generate pre-RNA. After that, they will make the complex. After that, they recruited the RDRC. So they recruited RDRC. So RDRC is composed of RDP1 and CID12 and HRR1. So RDP1 is RNA-dependent RNA polymerase. So this is not RNA polymerase 2. So in our multiple question, there was a question regarding this one. So in the RNAi-dependent heterochromatin formation, RNA-dependent RNA polymerase is working. Okay, so this is the RNA-dependent RNA polymerase. So they will synthesis double-stranded RNA is like this. After that, there is a function is like this. So finally, they will make the RITS structure like this. So and also we cover in our homework, we cover this one. So this is the experiment. So in nascent transcript model, so RITS with single-stranded RNA is bind to the nascent. Nascent means that newly synthesized. So newly synthesized transcript, after that, it will make the maturation and heterochromatin formation. So in homework, I asked to design the experiment to prove nascent transcript model. So we just combine with the artificial tailoring. So this is artificial tailoring to RITS. So artificial tailoring can bind to the mRNA. After that, we find that they will make the heterochromatin formation is here. Okay. Okay. So, and also finally, they will recruit CLRC. CLRC composed of CLR4 and something like this. After that, the CLR4 function as methyltransferase. It will induce maturation. And especially the CHP1, they decode the information, and also especially in the fission yeast, SUI6 from fission yeast and mammalian, especially the SUI6 can recognize the H3K9 maturation. So finally, they will initiate heterochromatin formation. Okay, so you don't have to memorize all of step, because this step is this RNAi dependent heterochromatin formation is unique from the fusion yeast, unique phenomenon from fusion yeast. So this is very important. So you don't have to memorize all of the steps, but you have to know some of the important step. Okay. Okay. Very important. Okay. Okay. And also there is difference between the function of RNAi in mammalian and also function of RNAi in fusion yeast. Especially in the mammalian, so they will make the risk complex in the cytosol. But however, in fusion yeast, they will make the complex. They will make the RITS complex in the nucleus. But finally, both of them, they will make the post-transcriptional gene silencing. Post-transcriptional gene silencing. But this is happened in the cytosol, and this is happened in the nucleus. And also, we learn the transcriptional gene silencing like this. Okay, and also we cover some maintenance of heterochromatin during cell cycle. So in midterm exam, so I will give you the picture. I will give you the two pictures. Okay, based on two pictures, you have to explain the phenomena. Okay, especially in G2 page and M page, and the G1 page is the like this. This is the lowest 3,6 binding status. That in the S page. So okay, so you have to explain the procedure very briefly. Okay, so in the M page, especially histone 3. So for example, histone 3 has lots of amino acid, especially the 10th amino acid is serine. So because some of you take the protein engineering course, so serine has they have OH group. So especially in OH group, the phosphorylation is happening in OH group. So phosphorylation of the H3S10, so phosphorylation of H3S10 lead to separation of 3,6 from methylated H3K9. That's why the G1 state is like this. And also during S phase, and lots of the event is happen. Okay, so this kind of event will be happen. But I just briefly summarize the event. So DNA replication happen. At the same time, the transcription happens. So finally, they will make the siRNA formation. So they will make siRNA. After that, dice will be happens. So finally, they will make the RITS complex. So it's like this. And after that, they will recruit the CLRC complex. In CLRC, they have the CLR4. So I just briefly summarize the function. Well, you don't have to write down the detailed information. Just you don't have to explain some important step. After that, CLR4 function as the methyl transferase. So it will make the maturation in the newly synthesized nucleus. So it can induce heterochromatin formation in both the pericentric heterochromatin region. Okay, this is the information because so I will show you the picture, but you have to explain the procedure. Okay, because this is also very important concept in chapter 3. Okay, and also there is the, so this is first step of the RNAi dependent heterochromatin formation. So, okay. So if you remove the exosome, so main function of the exosome, what is the main function of exosome? Their function is remove most of the non-coding RNA. It means that after we remove the exosome, it means that there will be loss of non-coding RNA. It means that Treatment can synthesize the more free RNA. It means that they will promote heterochromatin formation. How about another system is that, so this is the, actually, this is the single-strand RNA, okay? And pre-RNA is single-stranded RNA, and in the cell, in the nucleus, we have the mRNA export mechanism. Especially, they recognize single-stranded RNA that they export that RNA to the outside of the nucleus. So if we remove the mRNA exporter system, it means that pre-RNA stays longer in the nucleus, so it will promote heterochromatin formation. So this is the RNA quality control system and heterochromatin formation. Very important, okay? Very important, you have to memorize, okay? And we also learn about difference between constitutive and facultative heterochromatin formation, heterochromatin. Okay, so there was the multiple choice question was asked about this one. Okay, and also we learned about this one and also this one. Do you have any other question in chapter 3? Do you have any other question in chapter 3? Okay. Okay, so now we have to cover chapter 4 and chapter 5. So now we will go to the chapter 4 directly. Okay. So chapter 4 is the epigenetics in fruit fly. Now, do you have any question in homework or just the class material? Do you have any question? Chapter 4 is we cover in homework, we covered a lot of concepts. Do you have any question? Okay. Now we will cover epigenetics in fruit flies. Okay, so this is the fruit flies. Okay, they have 4 pairs of chromosomes. And chromosome 4 is very small, it's like this. Okay. And also in chapter 1, we cover position effect variegation. Okay, so in this case, if the allele W, last W is away from the heterochromatins, so they will make the red eyes. However, when allele W is near the heterochromatin lesion, so at that case, allele W is inactivated, but allele W is activated during some specific time point. So they will make the red eyes with height sectors like this. Okay. Now we cover the genes affecting PEV. First one is SU variegation. Another one is the E variegation. The difference between SU and E is that, so the very famous the SU variation means that surface of the variegation. It means that when a mutation occurs in SU, it will suppress the variegation. For example, the very famous SU is that H3K9 methyltransferase. 3.9 is H3K9 methyl transferase. It means that when a mutation occurs in the SU3-9, it means that there will be no H3K9 methylation. It means that there will be no heterochromatin spreading and formation. If there is no heterochromatin spreading and formation, it means that it will suppress variegation. Okay. Another case is E variegation. The 3 Gargar factor is belong to the E variegation. Okay, and also we cover the poly-10 chromosome, and also we cover the band. Okay, so in our homework we cover that the band region is just stained darker. So this is band region. This region is heterochromatin region. Interband region is the relaxed DNA and stained lighter. This is the euchromatin region. Okay, and also we cover TRXG and also polychrome gene. Okay. The main function of a TRX gene, okay, so main function in this case, some hox gene, for example, UBX is the a kind of hox gene, okay, so especially in this segment, UBX hox gene is expressed. So in this case, this UBX expression is maintained by TRXG gene. The main function of TRXG is maintain the active status of gene expression. However, polychrome gene, okay, so another segment, the polychrome gene plays important role in another gene. It means that in another segment, UBX protein is inhibited by polychrome PCG gene. So it means that this is the suppressed active status of, especially here, UBX gene expression. Okay, and also in polychrome gene, there is two complex, PCR1 and PCR2. Okay, and also we cover the 3 scenario: pre-dependent, or so there is a 3 pathway, 2 pre-dependent pathway, and also 1 pre-independent pathway. Okay, in our homework we covered like this. Okay, so in plot-wise, provide 3 possible scenarios of targeting mechanism of PRC1 and PRC2, the chromatin. Okay, there is 3 mechanisms like this. So in the actual exam, okay, so actually the PRC1, PRC2, they will make the facultative heterochromatin. Okay, not there is some difference between the facultative heterochromatin and constitutive heterochromatin. But PRC1 and PRC2, they will make the facultative heterochromatin. Okay, and there is 3 possible scenarios: pre-dependent, and also pre-independent pathway. Okay, so I will give you I will give you I will ask you one scenario. Okay, at that time I will show you the picture, and based on picture you have to explain the procedure. Okay, 2 pre-dependent, and pre-independent pathway. Okay. Okay, the main function, main complex of PCG1, they have a PC complex. PRC1 is composed of PC and SCE and PSC. Okay, especially the PC1 can recognize the maturation. How about the PRC2? They have the methyl transferase, and also another ESC, they can recognize the maturation. Okay. And there is a three pathway, three scenario is here. Okay. And also this is detailed information. Because the PRC2, they have the EGIT proteins. So it induced maturation. And the PRC2 and PRC1 they can recognize the maturation. So finally, they will make the facultative heterochromatin. It's like this. And also, especially in the fruit-wise, they have a PV, they have a PV siRNA. So this is the full name of PV is P element induced wimpy testis. It means that it occurs in germline, but sometimes they will also work in somatic cells. Okay. Okay, so this is the details information of the piRNA, and there was a multiple question asking about piRNA. Okay, and also this is the another important step in the chapter 4. In chapter 3, we cover RNAi dependent heterochromatin formation. How about in chapter 5? So we have the we learned about facultative heterochromatin formation by PRC1 and PRC2. Now we also have this one. piRNA and PTGS, post-transcriptional gene silencing. They have the composed primary piRNA pathway and secondary piRNA pathway, and finally they have they compose primary and secondary. Okay, so this you have to understand. Okay, you don't have to memorize the all over step. Okay, you don't have to, but I will give you some of the picture in the question, but you have to explain the procedure. For example, in these cases, so this is a primary piRNA pathway. So they have the procedure is like this. Okay, finally, HAMT3, they will make the maturation at the end, is like this. And this one, this is primary piRNA pathway, and this one will bind to the sense transcriptor of sense transcriptor. They bind to here. So they will cleaves the transcript. So this is this 5' to 3'. So this is 5' to 3', and this is 5' to 3'. So they will recognize the 3' fragment. They will make 2 fragments. This is the 5' fragment, and this is 3' fragment. Okay, so after like this, so final step is this one. This is the sense piRNA with RO3. Can recognize can bind to the antisense transcriptor of transposon. Okay. Okay, at that, this one can recognize, so at that they will make the fragment cleaves the transcript. At that, this one is recognized 3 fragment. This is 5' fragment and 3' fragment. At that, finally the antisense piRNA with OUB bind to the sense. At that, they will cleave the transcriptor. Okay, so it will induce post-transcriptional gene silencing. So at first step, they will also cleave the sense transcriptor of transposon. So it means that it also can induce PTGS, and also they also bind to the antisense transcriptor of transposon. So it will cleave the transcript. So it will also induce PTGS. And also finally, this one also can sense antisense RNA, piRNA with all AUB. They can bind to this one and they can induce PTGS. Okay, so I will give you the picture. So you have to explain, okay? You have to explain the step, okay? Okay. Okay, and there is also another PTG, transcriptional gene silencing pathway, okay? And also finally, this is a final subject in sex chromosome in plotwise. In plotwise, there is some specific mechanism, dosage compensation. For example, in the mammalian, we also have dosage compensation mechanism. For example, male has XY and female has XX. So in this case, in female, usually the random inactivation of one X chromosome happens. However, in the plotwise, the male is XY and the female is XX. So in this case, is the specific mechanism, X chromosome can be transcribed 2 times faster. So it means that the amount of transcript on synthesized on X chromosome will be doubled. So this is a specific mechanism. So we find that in this case, DCC complex is the function especially in the male X chromosome. So they have the H3K16 acetylation, and they also have the function helicase. Okay, they have another component is like this. The main function is to transcribe the X chromosome 2 times faster. There are several models, but most reliable model is model 4, is like this. So in the chromosome, maybe there is the high density of the DCC binding site, especially the CES chromosome entry site. So maybe the DCC is binding to particular sequence here, like this. So at that they will. So this is X chromosome. At that they will at that X chromosome can be transcribed 2 times faster because DCC is binding to the everywhere, nearly everywhere. At that amount of transcript synthesized on X chromosome will be doubled. Okay. Okay. We covered the important step. We covered everything. Okay. Do you have any other question in chapter 4? Okay, chapter 4. Okay. So chapter 4 homework, we already explained everything, okay? Course. Oh, okay. Okay. Okay, do you have any other question in chapter 4? Okay. How about now, let me go to the chapter 5, okay? Okay. How about we had better have 1 minute breaks, okay? Just 1 minute breaks, okay? So I need water, okay? Okay. Okay. Maybe some of them is the fourth year. 하여튼 다들 뭐 졸업할 준비 다 됐나요 이제? 오케이. 뭐 지금 다 진로를 다 결정 다 했죠, 맞죠? 본인이 다 진로 결정했으니까 그냥 본인이 결정한 진로에 대해서 후회하지 말고 열심히 이제 노력하시면 될 것 같아요. 이제 뭐든지 다 될 수 있으니까요. 오케이, Leah, do you have any other question in the class or chapter 1, chapter 2, chapter 3, and chapter 4? Do you have any question? Did you
Me? No, I think it's fine.
Okay, how about Monica?
It's fine too.
Okay. Tibor?
No, it's clear.
Okay, okay. And also I found it. Zana, Marcin Jana, do you have any other question?
No, it's good for me too.
Okay, because this epigenetics is very difficult to understand. Okay, so usually in the undergraduate course there is no epigenetics, but I opened this class. Okay. Okay. Marin, do you have any other question?
No, I'm good too.
Okay. 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 Now we will cover chapter 5. So do you have any other question in chapter 5 homework or the class? Do you have any other question? Okay. So let me explain the epigenetics in the mammals. Okay. So in chapter 1, we cover DNA maturation. Okay. DNMT1 and DNMT3A and 3B is involved in this step. So it means that DNA methylation means that cytosine, especially in the 5 carbon location, the methyl is added is like this. So they will make 5 methyl cytosine is like this. Okay, so there is 2 mechanisms. Okay. One is by DNMT1, the other one is by DNMT3A and 3B. Okay, so you can say like this. So this is the conservative DNA maturation. It means that recreates, this is the hemi-methylated DNA, but DNMT1 is recreates methylation pattern on the hemi-methylated DNA. Another mechanism is de novo DNA methylation. So they made a newly synthesized DNA methylation in both strands. Okay. And also we cover, so in our chapter 5 homework, so there was a question asking about this one. Okay. Okay. And also this is the maintenance model. Okay, like this. Okay, now also especially this is the structure of DNMT1 and DNMT3A and 3B, like this. So 3A, 3B, they have some specific domain, it's like this. And also in our homework, there was a multiplication asking about PWWP and ATRX, like this, okay? And also this is another mechanism. They have the cooperative activity, and also our multiplication, they ask about this active cooperative activity between the methyl transferase. So DNMT1 and 3A, 3B, they are cooperative to maintain DNA maturation. Okay, this is important bisulfide sequencing. So in chapter 5 homework, we cover bisulfide sequencing. Okay, so based on this is so this C is methylated, and also this C is methylated. How about this cytosine is not methylated? Okay, so write down the sequencing result of DNA after bisulfide treatment. Okay, it means that after bisulfide treatment, methylated, the cytosine still be the cytosine like this, still be cytosine like this. However, the cytosine, like just if they don't have cytosine methylation, so they will read it as the thymine. And thymine is like this. So this is the example. Okay. Before sequencing, DNA was treated with bisulfide like this. So it will convert cytosine to the uracil, but if there is the methylation is here, so it leaves the 5-methyl cytosine residue unaffected, is like this. So okay, so this after sequencing, okay, cytosine means that stands for methylated cytosine. Okay, if they are not methylated, they can be lead to the thymine like this. Okay, but if there is the C is leaded after sequencing, if we find the C, it means that C stands for the methylated cytosine. Okay, so this is methylation, bisulfide methylation. So you have to understand this one, okay? In midterm exam, you have to understand the bisulfide sequencing. Very important, okay? And also there is another transcriptional regulation by DNA methylation. Also another disease, Rett syndrome, is there is a mutation in the MECP2. So they will make some disorder like this, okay? And also this is the second subject is a DNA demethylation. Okay, DNA demethylation means that removal of methyl group from the nucleotide, especially the cytosine. Okay. So in the actual exam, I will give you the T structure, 5-methylcytosine. After that, you have to write down the structure of this one. So especially the TET, the full name of TET is 10-11 translocation gene. The full name of TGD is the thymine DNA glycosylase. Okay, the main function of TET is that they can oxidize, they can induce oxygenation in the methyl group. First, this is 5-methylcytosine. First, TET, they will make the 5-hydroxy-methylcytosine is like this, 5-HMC. After that, this OH is further oxygenated. So they will make the carbonyl group CO like this, double bond is like this. This is, we call it as 5-formylcytosine. So finally, TET, they will make a carboxyl group, more oxygenated. 5-carboxylcytosine is like this. At that TGD is this one is the they can remove this group. The main function of TGD, they have two functions. So they remove this 5-formyl group from the 5-formylcytosine, and also they have another function, remove the 5-carboxyl group. Okay, so finally they will make the cytosine is like this. So this is the DNA demethylation, the detailed mechanism of DNA demethylation. TT and TGD is involved in this step. Okay, the most important thing in the chapter 5 is the bisulfide sequencing and also DNA demethylation procedure. Okay. Okay, very important. Okay. Okay, and also this is the DNA demethylation and methylation during the development. Okay, from fertilization to blastocyst, and also another step is from the blastocyst and to the epiblast. Okay, especially the epiblast stem cell. And also another step is from the epiblast stem cell to the PGC. PGC is a primordial germ cell. After that, the methylation will be happens like this. Okay. So from the fertilization to the blastocyst, during this step, especially TT3, they did demethylation from fertilization to blastocyst. After that, from the blastocyst and epiblast, so demethylation is happened. After that, from blastocyst and to epiblast, so DNMT3A and 3B, not DNMT1. So DNMT3A and 3B, they did de novo methylation. And also, after that, from the epiblast to the primordial germ cell. So primordial germ cell is located here. The primordial germ cell. So also demethylation is happened. At that time, TT1 and TT2 involved in the demethylation step. Okay. So you have to know the DNA demethylation and methylation during development. Okay, you have to know. Okay. So pre-modular germ cell, so also undergoes the methylation. So finally, so there will be the babies will be burst like this. Okay. Okay. Demethylation, methylation, demethylation, methylation. Okay, important, very important. Okay. Now we will cover non-coding RNA, long coding RNA. So there is a short non-coding RNA. For example, microRNA, snow RNA, and snRNA, tRNA, and piRNA is short non-coding RNA. And also there is a long non-coding RNA. We call it as LNC RNA. So there is intergenic, intragenic, and enhancer RNAs like this. And also there is in the long coding RNA, there is another transcript, CUT and SUT. Okay, the difference between CUT is also belong to the long non-coding RNA. Okay, for example, in the multiple question, they asked about CUT is rapidly degraded by exosome, and also SUT is rapidly degraded by another protein is like this. Okay, but important thing is now in this subject we cover long non-coding RNA. Okay, especially the CUT and the SUT is a long non-coding RNA. And also there is a mechanism, transcription regulation by long non-coding RNA. There's two mechanisms, cis-acting and trans-acting. So in our homework, I asked about trans-acting mechanism. Okay. Okay, and also there is the transcriptional regulation by cis-acting long non-coding RNA, and also trans-acting long non-coding RNA. Especially, we can find the transcriptional regulation by trans-acting long non-coding RNA in the Hox gene cluster. Okay, Hox C gene is located in chromosome 12, and Hox D gene is located in chromosome 2. So usually the HOCTAR, this long non-coding RNA is located between the Hox gene cluster. So they will make this kind of cluster, and so this complex, they will induce methylation in Hox D gene in chromosome 2. So this is the trans-acting long non-coding RNA. Okay, and also there is we cover chromatin insulator in the mammals. There is a 3 scenario, barrier scenario, and also enhancer, blocker, and insulated domain. Okay, so this insulator is very important. Okay, very important for protein expression. Okay, so nowadays in the biopharmaceutical company, so they developed a vector platform to express the protein. So because in this case, they want to add the insulator, chromatin insulator. Because when the vector is integrated into the genome, at that time the vector expression, protein expression is affected by neighboring the chromosome. So if there is the chromatin insulator is in the vector, so it can prevent the heterochromatin spreading, or a lot of it can prevent this kind of functions like this. Okay, so nowadays, who is finding the chromatin insulator in the world? Who is doing the research to find the chromatin insulator? So it's me. I'm doing this research right now. So I just want to find the chromatin insulator region in the genome. Okay, after that, I just want to clone the chromatin insulator region and just want to put into our vector system. So if you are interested in this research, please contact me. Okay, I'm doing this research. Okay, and also another thing is CTCF. So this is a CTCF. This is also chromatin insulator in the mammalian. It's not found in the yeast, worms, and plants. So they have zinc ping domains. So this is the way how CTCF work. Very important. Okay. You have to know to solve the midterm exam. So when CTCF, then there will be two cases. One case is like this. One case is like this. So they will make the insulated region like this. Okay. So when this cases happen, so enhancer is promoter is they are located nearby. So it means that enhance interaction. So it means that it enhanced interaction between enhancer and promoter. It means that they will activate transcription. However, enhancer and promoter is separated by insulator, CTCF. In this case, it will block the interaction between the enhancer and the promoter. It means that it will inhibit transcription. So there will be two cases, okay? So you have to explain this mechanism. Okay, do you understand it? Okay. And we also covered TAD region, okay, topically associated domain, and finally we cover enhancer. Enhancer is as I mentioned earlier, enhancer is the cis-acting element to interact with promote region. Because it is almost impossible to find enhancer due to the lack of the motif. However, there is some motif is there. Okay, for example, enhancer region is enriched with the H3K27 acetylation. For example, this region looks like the enhancer because they have the P300. P300 is also histone acetyl transferase. So we can find that this region is highly enriched with P300. However, enhancer region is the lack with H3K43 methylation. H3K43 methylation. For example, based on these three, so they are highly enriched with H3K27 and also P300, but they are lack with H3K43 methylation. So we can assume that this region is the enhancer. So you have to understand this concept. Okay. In the actual exam, you have to find the enhancer region based on this data. Okay. Okay, and also there is another difference between the enhancer and promoter. And in our homework, we cover the difference between the enhancer and promoter. Okay. Do you have any other question in chapter 5? Okay. Okay. Do you have any other question? Okay. So we have next Friday, we have the exam. So so you had better attend the class around 10:25, around 10:25. Okay, don't be late because our exam will start 10:30. So from 10:30, you can download it midterm exam file. Okay. Okay, so from the 10:25, I will check it. Everybody, you are ready to take the exam? Because I will check your camera status. Okay. So if you are not ready, so we already submitted you guys already submitted your video. Okay. So, but if you are not ready to take the exam, I will not give you the chance to take the exam. I will not. Because if you do not show yourself, it means that I don't know whether you are doing cheating or not. I don't know. Okay, very important. Okay, this step, this one, and this one, and this one. Okay, and send it to Professor's phone or Professor's Kakao. Okay. Okay. Do you have any other question? Okay. Good luck. Okay. Good luck. See you next week. Okay. 신주환. 세명 해결.
네, 교수님. 예,
지난주에 안 들어왔던데 오늘도 늦게 들어왔네.
예, 맞습니다.
너무 해결 잘 이해했어? 어려울 텐데.
그래, 네, 열심히 따라가고 있습니다.
열심히 해야 돼, 알겠지?
예, 알겠습니다.
내가 개인적으로 몇 번 연락해서 열심히 하라고 했잖아.
예, 맞습니다.
Okay. Okay. You have any other question? Okay, so we will finish class. Okay, bye.
Bye.
감사합니다.
Bye. Thank you.